Good evening friends. Today we will discuss about patch clamp technique. Before that I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. Patch clamp is a laboratory technique used to study ionic currents in individual isolated living cells, tissue sections or patches of cell membrane. This technique is especially useful for the study of excitable cells such as neurons, cardiomyocytes, muscle fibers and pancreatic beta cell. This technique can also be applied for the study of bacterial ion channels in specially prepared giant spiroplast. Patch clamp technique was developed by Erwin Neher and Bert Sackman in 1976. For this work, they were awarded Nobel Prize in Physiology or Medicine in 1991. Patch clamp technique is based on a very simple idea. A glass pipette with a very small opening is used to make a tight contact with a small area or patch of cell membrane. A small amount of suction is then applied to the upper end of the pipette. As a result, the contact between pipette and the membrane becomes so tight that no ions can flow between them. So this is the patch of cell membrane which contains an ion channel. When the ion channel opens, then all ions flow into the pipette. This flow of ions from cell into the pipette gives rise to the ionic current. The ionic current is then converted into electric current, which is measured by ultra-sensitive electronic amplifier. Such an arrangement is called cell-attached patch clamp recording. In this figure, this is a patch pipette and this is an isolated living cell. This is the cell membrane which contains ion channels. This is the ion channels whose function we want to study. In order to form a cell-attached mode, a pipette tip is placed on the surface of the cell. This forms a low resistance shield or contact between pipette and the cell membrane. Then slight suction is applied at the upper end of the pipette. As a result, the contact between pipette and the membrane becomes so tight. Such a contact with a resistance in the range of gigaohms is called gigashield. In cell-attached mode, recordings are made from the membrane area which is associated with the pipette. In this case, the cell interior remains intact. The pipette is filled with a salt solution which is similar to the fluid present within the cell. Inside the pipette, there is a metal electrode. In addition, another electrode present in the bath solution serves as ground. Due to the formation of tight contact, this membrane patch becomes electrically isolated. As a result, when the ion channel opens, all ions flow from cytosol into the pipette. This flow of ions from cytosol into the pipette forms an ionic current which is then transduced into electric current by the metal electrode. The resulting current is then measured by an ultra-sensitive electronic amplifier. This is the basis of patch clamp technique. Patch clamping can be performed in two modes. One is voltage clamp mode and the other is current clamp mode. Most patch clamp measurements are performed using voltage clamp mode. In this case, the researchers clamp the cell membrane potential or voltage at a chosen value, and the resulting currents are then measured. This makes it possible to measure voltage specific activity of ion channels. Voltage gated channels are primarily studied in this mode. In current clamp mode, the electric current passing across the membrane is controlled and the resulting changes in voltage are recorded generally in the form of an action potential. Patch clamp technique is used to study ion channels in cells. A single ion channel conducts around 10 million ions per second. This technique allows researchers to study as how cells function normally and how disease affect them. This technique has many applications. Number one, patch clamping has been used for a long time for the study of neurophysiology. Number two, it is also used to screen drugs for their effects on ion channels. In addition, patch clamp help us to understand how disease develops due to ion channel malfunctioning. Along with calcium imaging, this technique has been used to study how calcium levels are controlled in the brain. Finally, patch clamp technique is also used to study electrophysiological properties and also to evaluate antiarrhythmic drugs. Friends, this is all about the principle and applications of patch clamp recording. For more updates, please subscribe my YouTube channel, Sam Biology Class.